Okay, proteins. Our next biological molecule. We talked about carbohydrates, we talked about lipids, and now on to proteins. So, what are proteins? Well, what are proteins in us? Basically, they are us. You're looking at proteins right now. I am proteins. Proteins are everything and everywhere. Everything, my fingernails are made up of proteins, my eyeballs are made out of proteins, my skin is made out of proteins. There are proteins everywhere. Proteins are the structural and functional part, basically, of our bodies. Our DNA codes for proteins. That's what our DNA is. The information that it holds is the information, essentially, for all the proteins in our body. For our body, that's how it controls us. Having said that, proteins are very important. Now, the basic building block of proteins is an amino acid. So remember, we have monomers and polymers. So the basic building block of a protein is an amino acid. And you can string amino acids together. You can have a protein with, what do I say here? So you can have a protein as short as 3 amino acids and up to hundreds and hundreds of amino acids. So you string these amino acids together, and those form a protein. So how does that work? Well, we'll get into protein synthesis later in a couple of units from now. So we won't go into the detail of that, but just know that the sequence of DNA, sequence of nucleotides in DNA, is what determines the sequence of amino acids in a protein. So let's talk about amino acids. What's an amino acid? So let's look here. So it's a organic molecule, so it has carbon and hydrogen, as all our amino acids have. And the structure of a general amino acid is you've got an amino group, it's called, or an amine group, which is basically a nitrogen. Remember, that's one of our atoms that you find in biological systems. So we have an amine group, which is just basically a nitrogen with two hydrogens bonded to a carbon, a central carbon, which has a hydrogen there, and then bonded to a carboxyl group. Carboxyl group, it's like an acid. That's why it's called an amino acid, because this hydrogen can pop off and make things acidic. But so we got a carboxyl group, and then this central carbon, and an amine group, and then we have this atom, this thing here that says R. Now, if you look on your periodic table, you won't find R on the periodic table. There is no atom that is R. R here represents the side chain of an amino acid. So every single amino acid will have an amino group, a carboxyl group, and this structure, this carbon. We'll have those three things. And what varies, there are 20 different amino acids. And the thing that varies between amino acids is this R group, this side chain. And the side chain is what gives amino acids, each of those 20 amino acids, their individual chemical properties. So they have different chemical properties, okay? So you can see the sequence of them is very important because you'll have different chemical properties. Remember I said shape is very important to biological systems because you'll have a different shape, a different chemical properties. Now, what do I mean by chemical properties? Well, I just mean chemical properties are things like whether something is polar, so it dissolves in water, whether it's non-polar, doesn't dissolve in water, if it has a negative charge or a positive charge. That's basically the sense. And then there's some like just size and shape. So here are the 20 different amino acids. So when I was in school, we had to in university we had to memorize these 20 different amino acids. You don't have to memorize them, but what you do have to know is that it's the R group, that side chain, that gives amino acids their unique. That was one of those 20 amino acids. It gives it its unique chemical properties. So that's the structure of an amino acid. And so then what happens is a protein will be a chain of these amino acids, and how they're joined up is all of these, you're always, remember dehydration synthesis. You're using dehydration synthesis, which is taking that water away to stitch monomers or smaller molecules into larger molecules. And so what happens is I guess you would cleave this OH and this H of an adjacent one and form a very strong, what's called a peptide bond. And it's a very strong covalent bond between these amino acids. So it'll be the amine group bonded to the carboxyl group of an adjacent amino acid, and the side chain hangs off the side. Is there an example of that here? Oh yeah, there it is. There it is. You've got to stitch these together. So there you can see the carboxyl group, the amine group, if you look in your notes, and the R groups hang down off the side. You lose a water, dehydration synthesis. And now you've made a dipeptide. So these are called peptide chains. Proteins are peptide chains. It's another way. I know the terminology is going to get confusing, but now you've got a dipeptide. Add another amino acid, you've got a tripeptide. And then add more amino acids, and you've made a polypeptide. A polypeptide is just a chain of amino acids. Okay? It's not a protein yet, actually. It's not a protein yet. It's just a chain of amino acids at this point. Now, I said proteins are important. Proteins do everything. So there's just backtrack a little bit. There's two major groups of proteins. There are structural proteins, and those are the proteins that function to make structures, to make things. So there are proteins in my fingernails that are structural proteins that make the structure of my fingernails. There are proteins in our cells that make the cytoskeleton of our cells, which is sort of like that holds our cells together. There's all kinds of protein in the hair, what hair I have left. The proteins there. And the other group of proteins is functional proteins. So these are proteins that do things. And so there's enzymes. Those are proteins. And enzymes, what they do is they catalyze reactions. We're going to have a whole unit on enzymes. Basically what they do is they're these molecular shapes that do stuff. They break bonds or they fuse bonds. So dehydration synthesis, stitching things together, is catalyzed by enzymes. There's an enzyme that comes along and does that. Hydrolysis, which is breaking those bonds, releasing energy, there's an enzyme that comes along and does that. You can see they're very important because that's happening in your body all the time. So there's all these enzymes doing that. Other functional proteins, so there's antibodies, which are part of your immune system. When we talk about the immune system, we'll talk about those. Transport proteins, those are proteins that are in your cell membrane and help move things, decide which things can move in and out of your cells. And hormones. So we talked about hormones, we talked about steroids. Those were lipid-based hormones, but there are also protein-based hormones. So you also have hormones that are protein-based. Remember, hormones amplify parts of your DNA. They amplify, or they can actually even silence parts of your DNA. The expression of your DNA. They alter the expression of your DNA. So very powerful, very important, because that DNA codes for proteins and enzymes, so you can turn down an enzyme or turn up an enzyme. So structural and functional proteins. So back to making these, we're at polypeptide chains. So we've got a peptide chain right now with these peptide bonds, these strong covalent bonds between the carboxyl group and the amine group of two adjacent amino acids, long string chain of them. This isn't a protein yet. So proteins have what we call levels of structure. And these are important because, remember, I said the shapes of things are important. The molecular shapes of things, the shapes, in order for enzymes to act on something, it has to have a specific shape. Enzymes work like a lock and key system. So in order for an enzyme to do something, to break, for example, a peptide bond, it has to recognize that carboxyl group and that amine group. They have to be in a certain shape so that it can break them. So we have these levels of structure. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary levels of structure. and basically they're like the levels of complexity. So if I were to use an analogy, let's say in a building, the primary level of structure would be just the materials that are used to make the building. The secondary level of structure would be how the frame of the building is assembled. The tertiary structure, let's say the tertiary structure would be how many frames or how many levels there are, and maybe the coating on the outside, the wood, the siding on the outside. And then the quaternary structure would be if there was more than one sort of section or more than one section of building. That's a terrible analogy. Let's just go through the levels of structure. Okay, so the primary level of structure, I'm going to use this as my peptide chain. I don't know if you guys can see this. Will it focus on this? It'll try. Oh, there it is. So this is going to be my peptide chain. And so the primary level of structure is just the sequence of amino acids. So each one of these little beads represents an amino acid. And so the primary level is just the sequence of amino acids. So, I've got, let's say, leucine here, guanine, cytosine, another leucine, and just that is the primary level of structure, is just the sequence of amino acids. Okay? That's it. The secondary level of structure, you can have two types of secondary structure. Something called an alpha helix, and then something called beta pleated sheets. And how that works is, let's just go in and look. Can we see? Oh, I did something. Is this a good? These images, have a look at these images, but let's just look at it here. Let's just look at an amino acid first again. Let's just look at that. So, amino group, carboxyl group. Now, imagine you've got a chain of these. You've got some hydrogens over here, some oxygens over here. You're gonna have an unequal distribution of electrons, remember? So you're gonna actually have some polar covalent bonds here, because you got these hydrogens here. These are gonna be a little bit positively charged. This is gonna be a little bit negatively charged. There's some polarity here. We're not talking in secondary structure, we're not talking about the side chains. Don't think of the side chains. The side chains are they're gonna play a little bit of a function here, but don't think about them yet. The side chains aren't involved here. It's just these amino groups and these carboxyl groups. So the amino groups and the carboxyl groups have polarity. And what ends up happening is when you have a chain of these, they will loop around each other, and they will form those weak hydrogen bonds, because this is positively charged, this is negatively charged, this is positively charged. So an adjacent amino acid will sort of flip around and form a weak hydrogen bond between its oxygen and hydrogen here. So what ends up happening is you have an alpha helix. That is the sort of the standard thing. And so what happens is every few amino acids, the chain curls around itself. And what's in between the sort of levels of curls, as we make the curls, I don't know if you can see them, so I'm making a kind of a helix, if it'll focus on me. Don't focus on my face, focus on that. Yeah, so making a helix, or I'm trying to, the best that I can, sort of, oh, here we go. If I do this, I can do that. It's not great. But you know what? In biology, it's also not perfect in biology. Biology never works perfect like this. So here's my, my alpha helix kind of thing. And you can maybe now we can show. You get the idea. Is it twists around itself? That's an alpha helix. It twists around itself, and if I scroll to it, you can see it makes a helix, sort of like a DNA molecule helix, but a DNA molecule is a double helix. This is just a single helix. And what's holding here are holding it together are these hydrogen bonds between the amine groups and the carboxyl groups of adjacent ones. Not directly adjacent. It's like it forms between the amino group and a peptide of nearby. So I just say nearby. We don't know how many. I don't know how often it is. So that's one type of secondary structure, is this alpha helix. So the peptide chain forms these alpha helixes. That's secondary structure. And some proteins, that's all they are. They're just an alpha helix. These would be structural proteins. So you have a tubulin, for example, inside your body, which helps make cilia and flagella. These tubulins are just these helixes of peptide chains. Okay? Excuse me. Now, another type, so you have got, I said there's two. So there's alpha helix, there's also beta pleated sheet. And what happens in a beta pleated sheet is, so you've got your primary structure, your chain of amino acids, and they're curling around happily making or maybe I can do it around my finger. They're making an alpha helix. But here, when it goes to make an alpha helix, one of these R chains, because we got 20 amino acids, we got lots of choices, is really big and clunky. So remember, the R chains are the things that hang off the side. And it doesn't fit. It can't fit into the alpha helix. So what happens is it makes a kind of a kink, and the chain kind of folds back on itself, and then maybe there's another one and another kind of thing. And so if I can do it, my rough demo is just, it sort of folds back and forth in on itself, the chain. So you can see here it's sort of going back and forth because of these R groups. Now the R groups aren't part of these, they're causing these kinks, but they're not part of this structure. Okay? Like they are, but they're not the key player, I guess. Because what's happening is then, as you can see these peptide, the kinks going back and forth, there's hydrogen bonds then that form that way and hold it together, hold this thing together in a sort of sheet. So you can see there's going to be hydrogen bonds going back and forth between these. And so therefore the other type of secondary structure is called a beta pleated sheets. They're pleated, they go back and forth. And you can see here they actually have, it's a three-dimensional structure. Will it focus on it? There we go. Is a beta pleated sheet. So you have one chain going that way, one chain going that way. They're going back and forth. And the R groups are sticking up and down off the beta pleated sheets. And again, and a peptide chain can have both. Some of it can be alpha helix and some of it can be beta pleated sheets all over the place. And those beta pleated sheets will basically make like these flat surfaces with R groups presented out. And the R groups, remember, are the functional groups. So you can have, if you're an enzyme and you got these things coming at you to do something, to react, those R groups can react with stuff. You'll see. Okay? I like that. So, secondary, so that's secondary structure. Then we move on to, and like I said, some proteins only have those two levels of structure. Tubulin, I used that example. There's also some of these ones, the ones that are like, what's that called? Keratin? I'm thinking keratin that makes up your fingernails and like hooves so those would be like more beta pleated sheet kind. So that's secondary structure. So all proteins have primary structure because they have to have a sequence of amino acids, and then all proteins have secondary structure because every protein has a carboxyl group and an amine group that will react with the carboxyl groups and the amine groups that are near it, and it will react either as a beta pleated sheet or as a alpha helix. And then we've got those R groups, right? And we looked at those. Let's just talk about, because tertiary structure is all about the R groups. And you can see there are all these combinations. Some of the R groups, like for glycine, it's just a hydrogen. Not much going on there. Some of them, it's, tryptophan, phenylalanine, they're like these ring structures. So there's a lot going on. There's amino groups, acid groups. There's all kinds of structures there, all kinds of atomic structures and molecular structures that are hanging off these R groups that give them these various chemical properties. And they can react in many different ways with each other. So what can happen is, so you've got your beta pleated sheets, let's say, and your alpha helix, and now the chemical properties of those R groups start reacting with each other and will change the shape, will form, those R groups will form bonds between themselves and pull the chain into new structures, more complex structures. I don't know if you can see the very complex structure that I've made. That's tertiary structure, when the R groups start reacting with each other. And you can, because they have so many, they're so varied, you can have covalent bonds form between them. You can have ionic bonds. Remember ionic bonds? You can have ionic bonds form between them, and you can have those hydrogen bonds form between them. It's covalent, ionic. You're going to have an enzyme. You're going to have something come in and make those bonds, do those bonds. It's not going to happen spontaneously. But the hydrogen bonds can form spontaneously, because hydrogen bonds are just that's like magnets coming together, a negative and a positive charge basically coming together. The majority of tertiary structure will be hydrogen bonds between the R groups. That's the majority. And remember, those are the weakest kind of bond. Covalent, super strong, but hydrogen is the weakest kind of bond. Remember, all this stuff plays constantly. So even that, when we looked in section A1, we talked about those bonds. Remember. So tertiary structure. Tertiary structure is complex, and it's what it will give the protein or its three-dimensional shape, a very complex three-dimensional shape, and its function. So if you're making an enzyme and you have an enzyme that has to act on something, so I don't know how and remember, enzymes work like as a lock and key. So you're making the lock, you're making the shape of the lock with those R groups. You're making a specific shape with the bonds that form between those R groups. You're folding the peptide chain into that. So if it doesn't have that shape, if it doesn't have that structure, it doesn't work. A lock wouldn't work if it didn't fit a key specifically. So the tertiary structure is obviously very important. Now, because it's made of hydrogen bonds, this structure can be broken. Hydrogen bonds are weak, remember. And basically what can happen is they're sensitive to pH. Remember, we talked about pH, very important thing. So basically, if there's too many hydrogen ions or hydroxyl ions in a solution, so where you have, remember, hydrogen bonds work, they're like magnets, right? Negative is attracted to positive. Well, loose hydrogens are going to have a positive charge, so they're going to start pulling on those hydrogen bonds. Same conversely for hydroxyl, they have a negative charge, and if you have too many, those bonds, there's too much noise basically, and wherever the bond was forming in the peptide chain, there's just too many things trying to pull it apart, and it loses that tertiary structure. The other thing is temperature. So temperature can break hydrogen bonds. Basically what temperature is, you would have learned this in grade 8, I hope. If I was your grade 8 teacher, you would have. Temperature is basically just kinetic energy, is the vibration of molecules, right? So imagine you have a hydrogen bond, like imagine I'm hydrogen bonded, I don't know, you guys can't see anything here, but imagine I'm hydrogen bonded to this desk. My arm represents sort of a weak hydrogen bond. And now we're adding temperature, so it's getting warmer and warmer, and I'm shaking and this desk is shaking, so that's all warm is just the molecules and the atoms vibrate more and spin more, and eventually there's so much temperature that you just can't hang on. That bond is too weak to hang on because these things are vibrating or moving too much. That's what happens. So hydrogen bonds and therefore proteins and enzymes are sensitive to temperature. They will denature. So they will denature means they will lose their shape if things become too hot or things become too acidic or things become too basic. That's why when you get sick, your body fights it with a fever. You get hotter because what your body is doing is trying to denature the proteins in bacteria and virus and bacteria proteins. Virus and bacteria proteins and enzymes are more sensitive to temperature than our proteins and enzymes. They have a thinner range that they can work in. That is a very good explanation. And then the last level of structure for proteins is quaternary structure. And all quaternary structure is you have one peptide chain, it has secondary structure, it has tertiary structure, and then a second, let's use this dongle thing, a second peptide chain comes along and interacts with the first peptide chain, and it'll be like the R groups interacting. They combine together, and that makes a large functional protein or enzyme. So quaternary structure is just other peptide chains with tertiary structure coming together and combining to make a functional protein. So an example of that is hemoglobin. Hemoglobin is a protein in the cell membrane of red blood cells, and it's 4 peptide chains that come together. They're called, and they make these things called hemes, where 4 oxygen atoms can bind, and that's how your red blood cells carry oxygen. Is in these in hemoglobin. Hemoglobin is the protein that carries oxygen. We'll learn about that in the circulation unit. So there you go. That's why you need to know all this stuff, because it's the basis of all these other things. So quaternary structure is just separate peptide chains. Tertiary structure is the R groups of a single peptide chain interacting together, forming covalent bonds, ionic bonds, hydrogen bonds, mainly hydrogen bonds, to give it structure, that gives it function. Secondary structure is the carboxyl and the amine groups of adjacent amino acids hydrogen bonding together to form either beta pleated sheets or an alpha helix. And finally, primary structure is just the chain of amino acids, is that peptide chain, the individual, the order of amino acids. What is the order of amino acids? And you can see that comes in, the order of amino acids becomes very important for tertiary structure, which is the functional sort of structure of proteins. Is there anything else? No, that's it. Nucleic acids next. So there you go. That is proteins.